Hello and welcome. In this series, we continue to talk with the scientists of the Dangoor Center for Personalized Medicine. Get to know a little bit more about them, what's happening in their research laboratories, what developments are expected to reach the market, and what medicine will look like in the coming years. This time, we're very fortunate to host Dr. Sharon Gilaie-Dotan from the School of Optometry and Vision Science at the Minna and Everett Goodman Faculty of Life Sciences. She's also associated with the Ganda Brain Research Center in Bar-Ilan and, of course, the Dangoor Center for Personalized Medicine. In her lab, Dr. Gilaie-Dotan researches the visual system in the brain and how it's related to our visual functioning and our behavior. Dr. Gilaie-Dotan has a bachelor's, a master's degree in mathematics and computer science from the Weizmann Institute. She continued her studies there for her doctorate, but in neurobiology. And she did a postdoctorate at University College London, and in 2016, she joined us at Bar-Ilan. So, Dr. Gilaie-Dotan, Sharon, we have the same name, so yes. I can call you that. Tell us a little bit about the visual system and how it relates to the brain. We all know the eye, but clearly your work, you're associated with the Brain Center. How does it all work?
Yeah, so we've got many visual functions, and the visual system is what allows us to view the world. Of course, we've got the eye and the muscles that move the eye, so it's not only a sensory system, it's actually also a motor. It has motor components where we move, we adjust the optics and we move our eyes across the visual space in order to see different things. But beyond the eye, which is a very complicated organ on its own, we have many visual regions actually in the human brain that support our visual perception and our visual behavior. We have we have dozens of regions actually within the visual system, which is actually here at the back of the brain. Some of them have a replication of the visual image in the retina. So they basically have maps. We have multiple maps of the retinal image, but beyond that, we have a specialization with regions that are more sensitive to different things that are more closely related to what we understand. So, for example, regions that are sensitive to faces over other things, regions that are sensitive to body parts, regions that are sensitive to color, regions that are sensitive to places, to motion, to disparity. Disparity is what we would say, stereo vision, the differences in so we have many types of specializations, and we can actually split them or categorize them into two main functional families. Those that allow us to understand what there is in the world, and those are located like down here, like close to where the ear is, you would say. And this is called the what stream or the perception stream, and it allows us to understand what's around us. But since we are dynamic creatures, we move in the world, we're not, we plan our actions. There are additional family of functions that is more associated with where things are in the world and how the space around us is organized. And this is more upward here, kind of like anatomically connecting between the visual system and the motor and sensory systems. Basically, it's assumed to support our planning for actions across, when we have to move ourselves in the world, because we think of vision as supporting our perception of the world, but in fact, a very big role it also has in allowing us to move in the environment safely and to keep our body safe. We don't always move in the shortest spatial distance.
Right.
Most importantly, we keep our body safe, and vision has a big role in that, allowing us to do this without getting our body harmed. Yeah.
So I think part of my brain works well because I can get the what, but I'm constantly bumping into things. So you're saying to me it's different parts of my brain. Some may be working better than others. But that leads to my next question, because this is the Dangoor Center for Personalized Medicine, right? So does my brain, my visual system work differently than your visual system? And can we adjust things differently? Or, how does that work? What makes it personalized?
Yeah, okay, so of course we have many commonalities. You know, both of us have eyeballs, and both of us have areas in the back of our brains, and you know, the wiring is very similar, but there are many, many differences, starting from, you know, basically the optics, you know, and some people require, you know, optical corrections, because even our eyeballs are not, you know, uniformly anatomically formed. And we know from the retina that some people have even differences. We've got 3 types of photoreceptors that are sensitive to different light waves, and this is basically what underlies or supports our color vision. But there are people who have deficits in color vision or have non-typical color vision, and most often this is caused by the fact that they either don't have these 3 types of photoreceptors, but only, for example, 2 of them, or some people have 3, but 1 or 2 of them have different sensitivities, so their color perception would be skewed. Different, you know, and this is something that we see, and this is already from the retina. We also, you know, in the retina, we also have differences in contrast sensitivity. These things can change across our lifetime. So many people, as they age, they start complaining that they need more light in order to read and stuff like that. So we are also not constant, you know, across our lifetime. Visual system matures, then reaches some plateau, and then starts to, you know, change as we age. But we also have, in what we call high-level visual functions, very big differences. For example, face perception, which is something that we in my lab also investigates. And we have people, we are all, I say, you know, on the spectrum of face perception abilities. So some people have deficits. They don't register a face. So while they understand they're talking to a person, they can understand, you know, many things about the face, they don't remember it. So if they were to talk to you, they would walk out of the room, they would come back, and you would change something in your hair. They may not understand, or there would be more people, they may not be able to find you and also their family members. Oh. Yeah, so it's very,
That's very traumatic.
It's very dramatic. They do recognize people, you know, using different types of cues, such as their voice, their body movements, and, you know, associations where they're expecting to find them, and they know them, but not through their faces. And on the other end of the spectrum, at the other end of the spectrum, we have people who are what we call super recognizers, and that's people who basically cannot forget a face. This is what they say, you know, they would remember a person they met years ago at some store, you know, these things that we would usually not remember. So, and all of us are probably somewhere along this continuum. So, and also people have differences in spatial navigation, which we tend to think may not be that visual, but it's also very visual. It's a very visual skill. Also, people vary in, so most of us, when we see black text over white, you know, paper, that's what we see, but there are conditions that are, again, recently known, called as synesthesia, with some people experience, you know, specific letters or numbers in various colors. So when they see that, you know, the color pops, they can't help it. It's not something that they can control. That's something that is different probably in the way their different areas in their brain, the visual system, are wired. So there's definitely many variations, also in stereo vision and in sensitivity to many other types of information. And it is important for us, you know, to understand these differences in order to provide, you know, corrective treatments or practices, or in order to understand, so each of us, you know, we know that, for example, some of us are better in reading, some of us are better in math, some of us are better in cooking or navigation, but once we understand that the visual system is composed of so many different functionalities, and some of us can be better in some aspects and worse in others, we can try to target and provide more comprehensive, individuated care
fascinating.
Yeah, it is
I mean, I
fascinating.
never realized I have glasses, so that's what I think of with visual correction, right? I have astigmatism, very bad vision, but what you're talking about is actually much more interesting than simply correcting the lenses. It's all brain processing.
So it's this and that. So I mean, if you wouldn't have your glasses, you wouldn't be able to perceive the world, you know, the best you can.
I wouldn't be able to see you. So forget the world. I'm pretty bad vision, but yes.
But you would want to correct the inputs.
Yes.
But also after the inputs are fine, you also want to see that the functions are doing the best they can, and so we want to see for each person how they are, their complete, let's say, profile. And we actually don't know what this complete profile is yet. This is something that we're trying to understand now, because most of what we know about vision comes from assessing vision at the center of the visual field, usually when people are sitting down and they're instructed to do things. We don't know a lot about the peripheral vision and what happens to vision when we move around the environment, and that's a very big part of what vision actually provides us with. So there are many things that we are still, that we have still yet to find out.
But what do you focus on specifically? It sounds like there are,
yes.
Who knew? But now I know, thank you. But there's so many different areas of the visual system you could be looking at. What do you look at in your laboratory? What excites you?
Yeah, okay. So vision excites me, to be honest, to begin with, but really our lab now focuses, I mean, most of the knowledge we have is about information that has been acquired, and extensive information from the center of the visual field, center of the visual field, when usually people are seated, so I'm saying seated because this is a situation where your body is safe, you're not likely to bump into things.
Just to be clear, center of visual field means literally the center when I'm looking straight at the center.
Yeah, so yeah, it's a very good question. What is a center? But because we have the point in space where we fixate at each different time point. But you can think about the area that we know a lot about. I mean, if you would just, hold this, and so this would be more or less the area in the visual field that we know a lot about. But it's a very small area compared to our experience. We've got a lot of information, and think of yourself as having to move in the world when this is the only visual information that you have. This is quite annoying.
For sure.
We rely a lot on our peripheral vision, but we don't have a lot of knowledge about that and how people use it. I mean, maybe it's not that much in our awareness to begin with.
But it would be like driving without using our side mirrors.
Much worse. You won't be able to see, the whole front glass, the whole
just
front
like a
window.
little tiny part of it,
yes,
right?
just a tiny part of it, and that's not something, that's not safe. You wouldn't feel secure. You wouldn't probably.
I wouldn't drive.
Yeah, you
wouldn't.
You would
drive.
just exactly. You would turn
I'd still
off the car.
stay in, but just sit there. Correct, but we don't function that way. So
yes, exactly. And many of the things that we know are from people who are, at the age again when vision has already matured, so 18 to 35. And before vision started to change. So we know, and it's from, so I said people who are seated, and usually people are told what to do about the stimulus, and they're usually looking at things that are at fixed distance away from them in flat screens, but just even here and anywhere, things are at different distances, and usually nobody tells you what to do with your vision. You just, we both may be looking at something, and you may be, focusing on it, and I may be wondering, my mind may be wondering about, thinking about other things. So we try to investigate, try to go beyond these, I call them, the experimental comfort zone. We investigate things in the periphery. We have exciting results. We don't go that far into the periphery yet because, we have our experimental limitations, but we look how peripheral information is processed relative to how central information is. We have a few papers about that, both perceptually, and also we also investigate how that influences regions in the brain that were thought to process, let's say, faces or places, but they were only tested or examined when information was at the center of the visual field, and we find that they, their profile kind of like changes when we look at what happens to them when they are exposed to information that is not in the center. We see that people, for example, this is a paper that just came out, are better at recognizing positive, like happiness, emotions in peripheral vision, and much worse at understanding negative emotions.
That's a lovely thought. There's something that says about human nature. I don't know exactly what, but it's making me happy. So
yeah, it was. I mean, it is surprising because this was not something that was clearly, predictive.
Right.
It wasn't easy to. I mean, we know that that's something that is happening in the center of the visual field, but it wasn't clear what's going to be happening at the periphery. Maybe we won't be able to.
And you would think, evolutionary-wise, we should, from our peripheral vision, be able to recognize danger, recognize anger.
Exactly.
It would be a warning, but you're saying no.
Yeah, there are
That's fascinating.
It is fascinating, and there are a few potential explanations. It's not that we know how this came to be. Actually, there was a fascinating study in Science examining behavior of macaque monkeys on a isolated island, and they're very aggressive. And following some disaster, shade, many of the trees where there was no shade on the island, and researchers examined how their behavior changed. And surprisingly, they became much more friendly and patient with each other. And actually, the ones that were the friendliest and the most tolerant to others were the ones that had highest survival rates. So I know that we can think that it can go both ways, but we found this, and we also see that face perception deteriorates much more than perception of places in periphery, which also makes sense because places are usually in the peripheral vision. And so this is part of what we examine both in processing and in peripheral vision. We also examine how different types of visual information influence our processing or our perception when we don't tell people what to do. We just tell them to view the images, which is something that is typically not done, and then we investigate how that influences their knowledge and processing later on. We also investigate visual information, because what happens when people move around? What are the inputs? Are the inputs just uniform, or so we investigate that, and we actually found that information in the lower part of visual space is closer to the eyes than information in the upper part of visual space. And we also found that people, although we think you can estimate whether this glass or this glass is closer to you, we found that we think we do this pretty good, even though you don't know maybe to say if this is 1 meter away or 5 meters away, you would know to judge what's closer to you and what's further away. But we find that people are biased to say that things that are, even when they're the same distance, things that are lower are closer to you. And so we have these biases, and we think this is related to the statistics of the information that we have, acquired throughout our lifetime. So also investigating visual inputs and understanding that this is, a very complex and diverse, stream of information that keeps on coming, as long as we're awake.
Right.
It also determines how our internal representations are, and it somehow, in some way, dictates our behavior. So that's
just a few of the things you're looking at.
Yeah, and we also and also we look into VR, because in VR you can have.
I was going to ask you about technologies, right? There's so many advances.
So many advances.
So what do you use in your laboratory, and how does that tie in? Things must have changed over the last 20 years, 10 years. I don't even know when these things. VR, the MRI, the fMRI, all of these advanced. How do you use some of that in your research?
Yes, so we have, of course, we investigate perception, and we also, as I said, record visual inputs of people moving around, which is, and then we analyze it. We examine performance not only through what people report, but also through their eye movements, to see if our eye movements can be telling or informative about, and predictive even of what is going to then be their decisions, or what they're going, because eye movements actually guide us. You can think about that, when you want to, grasp something or move, usually the eyes would lead and then our body would follow. And so, for example, in the VR environment, we can have, depth and information very similar, not precisely, but very similar to our natural environment. But yet we have measures, and we can control that. We can control the information. We can measure where things are and how distant they are from the eyes and from the hands, and we have, which is something that is very difficult to do when people just move around the environment, which is not really that controlled. We investigate things with fMRI, and you're right. This is, it's an amazing invention. That is definitely for medical purposes, but we also use that to understand and to learn about brain functionality and visual system functionality, and we do that. We don't have an MRI in Bar-Ilan, but we do that with other centers who provide who do have fMRI facilities. We collaborate with Professor Meir Plotnik in Sheba Medical Center, which has an amazing facility there, rehabilitation for walking, and we investigate there both in elderly, in young people, people with Parkinson's disease, the influences of vision on motor, and how, and thinking about how treatment can be, how we can provide and develop treatment. So yeah, it's not the only thing. We also have eye tracking in the wild, and when people walk, you can track where they're looking at.
In the wild, I love it.
Eye
tracking in the wild. I'm picturing like panthers, but you mean people walking down the street? That's great.
Yeah, in the wild, and science is outside the lab, basically.
Right,
That's,
I know, but it's just a great term.
Yeah, but yeah. So in a more naturalistic environment, so we try to use all these technologies in order to, to learn more about vision, daily vision, because the lab is not always, while we can obtain a lot of information about that, it's not always enough to understand how people function in the lab. Sometimes you need the more naturalistic environment in order to obtain much more knowledge and observations.
So you said you work with Sheba, you have a collaboration there.
Yes, definitely.
Which, by the way, this is so interesting because maybe I'm the only one, but I think most people, when we think of physical rehabilitation, we really don't think how important the visual system is. And it's wonderful that you're bringing this knowledge out there, and it obviously makes a huge difference. Logically, it makes sense. But when you think medically, therapeutically, do you have other collaborations with other people?
Yes, we have it in the, we have multiple collaborations. One of them is with Professor Boaz Ben David from the Reichman University, where he investigates what happens to our cognitive abilities during aging, and we are looking into results that we have in the lab. Cognitive studies, visual cognitive studies in young individuals, and we look at what happens in aging, and this, we think this can be very informative to see if the things that influence vision when people are young are, what happens during aging, and whether we can improve, facilitate, what are the factors that can, provide visual information in a more effective way, addressing the, potential difficulties, because people have, an assortment of complaints and difficulties that they report on when they age. Some of them are related to vision, some are related to memory, through cognitive functions, and we try to understand what happens, what we investigated, how that, in young people, how that changes across the lifespan. And of course, I really have to say that vision is not, it's not a standalone system. It's, we were talking about how it's, works with motor system, but it also works with audition and, with everything. So it's associated with all these different aspects, is influenced by and influencing, other aspects of our being. So we also have to, take that into account.
Sure.
And collaborations. We also have a collaboration with Professor Ifat Levy from Yale University.
So it's science. Yeah. You're doing wonderful things and working with other people. And as you said, because the visual system is so connected to so many of the other systems, it's important to work with experts in those fields.
Yes.
But speaking of crossover, you have an undergraduate degree and a master's degree in computational science and in mathematics, and then you went into neurobiology.
Yes.
Is there a connection? Do you use, you said you use virtual reality, you use an fMRI. Do you use your computational science skills in your research?
So yes, definitely we use computational, computational knowledge. And today, I think it's really, it's a step forward if we can rely on computational abilities, even computational models, but on, analyses that we can do with computers. They can take us much further beyond to understand very different aspects of the data that we may be not even looking at if we're just looking at kind of like more manually. So definitely we're relying on that, and but it's not that that's the only thing that counts, I would say. But definitely that does come into play when we work in the lab.
So you bring that advantage. You get to look at much larger amounts of information and use AI to process it and understand things that you might not be able to do even as a brilliant researcher, because you're human.
Yeah, there are many, there are tons of tools available, and now just as we speak, there are all these amazing, chatbots which, which even create code for people who don't have a lot of knowledge about, coding. So this is, we are in a very fast forward, times, I don't know what, not 30, but we're moving really, really quickly toward an era which we don't know how it's going to evolve, but definitely many people who have not been using computational skills maybe now be able to rely on tools that weren't, available to them. It's going to make coding much easier, but definitely there are many, many developments, and we try to keep up with them, but also I always think that we have to rely on our intuition and think about our experience as human beings and not as machines, to feed into the logic of what we want to investigate and what pieces of knowledge are still missing.
Well, you need to feed the questions into the machines, right?
Yes.
You can't. They don't just spit out information. You have to ask the right questions. You have to ask them in the right way. You have to think through what to ask, basically.
And you also have to be, this is also something that we have to be critical about the outputs that they provide. Correct. Because things that we do from scratch, we are more in control of, and we have some...
See all the processes and the kind of the thought processes here.
Thought processes, and we can, be responsible for the all the chain of events, but we don't know how the machine provided these outputs, so we have to be critical and to test them and to see that they're really doing what we're asking them to do. But definitely this is a brilliant advance in,
So I asked you the opposite, but now I'm going to ask you, what led you to go from computer science and mathematics into neurobiology? I mean, some of the conversation is because it clearly excites you, it interests you, and as much as you appreciate the computational stuff, you kind of like people. But tell me really what your thought processes were when you went through that professional transition.
Yeah, so when I finished my first degree, I went, I was working as a, programmer.
High tech. You're Israeli.
Yeah.
You went into the startup nation.
Exactly. And it's interesting that my grandfather, who was always telling me, you have to, get an education, be independent and all that. He's like my mentor. He actually was devastated by the fact that I'm not, okay, you're working, that's great, but aren't you interested in learning more and, doing higher degrees? And I was quite surprised. And that I thought, okay, well, I'm going to go and do some, second degree. Let's see how that goes. And, I love math. So that was...
I don't hear that often.
But that was, that was fun. But then I took this course, Advanced Topics in Brain Science or something like that, by Professor Barash in the Weizmann, and that course just blew me away. It was like, there was, all these phenomena about like left side neglect, where people neglect the left side of their space, sometimes their body, and I was like, what? How could that even be? I mean, really? I mean, I always knew we had a brain, but I didn't know what it did. Nothing. It's just, me and biology.
It helped you compute math. That's what it did.
Exactly. Yeah, I didn't even think. Yeah, exactly. And even my grandmother was always saying, use your brains, not power.
Right.
But I didn't. It didn't register to understand that there are, maps and that this has how this actually works and is organized. And I thought, okay, this is really interesting, but I have no background in biology, so I went and talked to Professor Rafi Malach in Weizmann, who had an amazing also a PhD student, Kalani Grill-Spector, who's now a professor in Stanford, and they, I talked to them and I decided, okay, this is where I want to be. And then I talked to Professor Shimon Ullman, who was in computer science, and then I had this joint supervision by both Rafi and Shimon, and I joined Vision, and I can say, we think of, so just to give you a flavor of what you're asking, computation, and how did I get to neurobiology? So, finding out that, the visual space that we see, you can think about it as a picture, you take a photo with a phone, and, we think of things as, having some X and Y coordinates, X, but the brain doesn't process the information that way, or it doesn't represent it that way. The presentation is what's called polar, and it's polar. So every point is not by its X and Y, it's by the distance from the center and the angle that it forms. So it's another, so it's a different mathematical transformation, also with a log, representing the fact that we have much more representation of the center of visual space. And for me as a mathematician to realize that, the representation in, our visual cortex is based on a mathematical, representation that is not, not straightforward. Yeah.
Taught,
it was
right?
Just, wow, it's amazing. Within a biological system, we have this, maps of, log polar representation of the visual space that was just like, yeah, it's crazy. And we see that in, in different species and, different systems. Also, we see this in motor cortex and sensory cortex. So this is just, it's amazing. So that's how
So you never left math. You just are applying it, and you had a whole new field of mathematics to explore within the brain.
I don't do math, but
know,
definitely I use it, and it's great.
That's fascinating. So, but you do take all kinds of multidisciplinary kind of perspectives as you do your research. So as somebody who's looking from all different angles, where are we going? Where are we going with all of this? If I were to say to you 5 years, 10 years, where are we going with this, both in terms of research, in terms of developments, and maybe most critically for most people, VR's fun, but medically, where are we going to take some of these things? Yeah,
so I really think that, now with the mass computational aspects and, the information, I think there are, substrates and foundations to have to keep much more knowledge and to look at much wider aspects. Understand that vision is much more complex, but not only vision. And I'm hoping that this information will be able to be utilized into really doing dedicated treatments, restorations, care. For example, a student of mine who's actually now graduating, finishing his PhD, he examined data collected. Bar-Ilan does, we have a School of Optometry, we have clinics where we go out to the public and we do this.
You have a mobile van even that travels to the periphery.
Exactly, you know about that.
Of course I know about it.
Exactly, so with all these, volunteering work to reach people who don't have either the means or awareness of the fact that they can improve or test their vision. We do this, and he relied on information that was gathered from a class of first graders where they were just starting to read, and he examined their screening or the visual examination results and the reading tests, all anonymized, but still, he was blind. And he found that about a third of the class had issues with their ability to control their eye movements. So this is something that is, it's disregarded. Usually kids are estimated, are examined for their visual acuity, which is clearly important, but there are many aspects more. And so, if we could use this information, again, of course, this has to be replicated and to see, but if we can use such knowledge and, improve our care, think about these kids, if they would have some kind of treatment, or not even medical, even behavioral treatment to improve their ability to control their eye movements, how much that could potentially affect their scholastic skills. So we can use all these different types of knowledges to basically treat people in a more dedicated and individuated way. And I'm hoping that, in the future, you talked about all these amazing technologies that are coming up. At the moment, all these amazing technologies are not necessarily individuated. You've got all these, high-tech glasses and stuff like that. So these are new and different products, but sometimes they're not individuated to your own first of all, to what you're doing. If you are walking, if you are sitting, they work, they don't adjust to the specific visual tasks that you are currently doing. And they don't necessarily adjust to your own specific abilities, how your eyes team together, if you do or you don't have different issues with countersensitivity, if you do or you don't need optical correction, if you do or you don't have issues with recognizing people or environments. So taking all of these things together, or colors, and incorporating that into all these new technologies, making them not only amazing, but individually amazing. That would be, individually tailored. That would really
life-changing, life-altering.
Life-changing for many people. For many people.
And what you've highlighted, which I never thought about, is how much the visual system and corrections to the visual system could make an enormous difference in so many spaces, whether it's first graders learning to read, which has incredible consequences for their academic success, to whether an elderly person can have physical rehabilitation, to across the board. It's extraordinary what some of these advances that you're working on could really transform so many people's lives.
Yes, me and of course additional labs. Yeah, of course.
Okay.
Yes,
but I'm sitting with you, so I get to give you the credit.
Definitely. So knowledge, I think, is the key. First of all, to have the knowledge, then you can decide or think how to use it in order to create devices or treatments to improve vision.
So how did Bar-Ilan get lucky enough to get you back here? You had this great postdoc. How did you get pulled back to Israel and to Bar-Ilan?
So first of all, it was clear to us that we're coming back to Israel. This is our home. And it was just when Bar-Ilan was, the School of Optometry and Vision Science was establishing as a new school with a research team. So there was an optometry department, but it wasn't, there wasn't much research going on. They basically established a school and amazing researchers with the great, the Faculty of Life Sciences, and with the Dangoor Center. It was clear to me that I want to be part of, building this school. It was great with the clinics, with the optometry clinics, there's so much knowledge about vision from aspects that I wasn't really familiar with, you know, all the clinical aspects and, you know, issues, you know, from neuroscientists usually, you know, treat the eye as some, you know, that's the input. That's like, you know, you've got to breathe, you've got to have the eyes. But we see that the eyes are also, you know, they're part of the visual system, and you know, they also have a big contribution, and so it's really fascinating also to think about them as an integral part of the system, not just some input facility. So definitely it was the best place for me, and it was, you know, what I wanted to do, so it was very natural to join.
So thank you for joining me today. This was really eye-opening, excuse the pun, and enlightening. Goodness, seems like all those words are
yes,
a little,
definitely.
You know, but really it was wonderful to hear about the extraordinary work that's being done with the visual system that your lab is working on, and to get a broader sense of the impact of this field. I appreciate you being here.
Thank you so much for inviting me. It was a pleasure to be here.
Thank you.
You.